Coming to our cozy third STEM diversity social. We have 4 speakers today from UC Santa Cruz, so we are fortunate to have 4 speakers. First up is Pamela Watson, and she's going to speak to you about her personal and professional experiences as a PhD student in the Molecular Cell and Developmental Bio program at UCSC. So I'll welcome her.
Thank you for the intro. So I'm going to start off giving a little bit of very basal level background information about what we do in our lab. Then I'll tell you a little bit about how I got there and what it means to be a PhD student. So I work in a lab, although I'm an MCD bio, so molecular cell and developmental bio student, I work in environmental toxicology laboratory. And what we do in our lab is, well, the main theme of our lab is to understand how microbes contribute to, excuse me, I don't know what to do here.
Move the mouse.
No, it's not the mouse. If I turn it right here.
I don't know. Yeah, it's the hot ones now. Oh, there you go. Right? Yeah. Cool thing.
Great. Thanks. So, yeah. So what we do in our lab is try to understand how microbes contribute to the mobilization of toxins, metals, and other natural minerals and such. So pretty much the way minerals move, there are some anthropogenic causes of the mobilization of toxins. So what I'm talking about is toxins that are naturally found, such as arsenic is one of our favorites in lab. We also work with chromium as well. So there are some things that we do as people in industry that cause the mobilization of arsenic. And there are some things that naturally occur. And then also there's some microbial processes that contribute to this mobilization. And the reason why we care about this so much is because once a toxic metal like arsenic, which was naturally found bound to minerals, gets liberated and starts moving around, it can end up into our water supplies, to our food, and then to interact with and inhibit our daily lives. So what we do more specifically is we try to understand the genetics behind how microbes can move arsenic-5, which is one species of arsenic. So what we've learned so far in the Saltipov lab is that some microbes can go through dissimilatory reduction, which is their form of breathing for energy. So instead of using oxygen when there's anaerobic conditions, so no oxygen and arsenic around, and naturally occurring minerals, these microbes use the arsenic and then it can get converted into its more potent toxin form, arsenic-3. And this arsenic-3 is water soluble, so as I was saying before, once these microbial processes affect how arsenic is bound to the mineral, it can end up in our water and such. So what I do is take a genetic approach to understanding how this happens. So I work with the microbe Schuanella species Anna3, and a lot of this stuff is what we all do, so you're going to hear the repeated themes here also. So I work with Schuanella species Anna3. It is a facultative anaerobe, so it uses oxygen, as I was talking about before, for respiration, and it can also use arsenic, chromium, and some other metals when there's no oxygen present. And what I want to do here is understand the genetics and what cellular processes lead to the use of arsenic as a terminal electron receptor for respiration. So what we know about arsenic so far is that there are several gene classes that contribute to arsenic reduction. So what I have here is a schematic of each arrow represents a separate gene, and these genes are classified into groups, where we have the one that I'm mostly focused on are the A and B genes, and these are the ones who directly are involved in respiratory arsenic reduction. There are some other genes here too that are important for preventing toxicity of arsenic, because it is also toxic to microbes as well. But what I focus on are the ARR, AB genes. And what I want to understand with my project is how are these genes being regulated. So most recently, I found that the cyclic nucleoside, cyclic AMP, is really important for turning on the genes that are important for arsenic respiration. So cyclic AMP is found in all organisms, including us, and in humans and most eukaryotes, it is a second messenger, and in microbes, it can actually act directly with the cyclic AMP protein to actually do genetic regulation, so turning genes on and off in the presence of different substrates. So what we think is happening is cyclic AMP itself is somehow gauging whether there's arsenic or oxygen present and turning on the genes that are important for arsenic reduction. And what we hope is, if we can understand how this happens, we can kind of think of and engineer ways of controlling the turning on and off of the arsenic reduction genes and prevent the microbes from reducing arsenic into our environments. So a little bit about me. I am actually a first generation Haitian American student. I had 7 siblings, and we were originally in, well, this is my mom here. We came from Orange, New Jersey, and what I wanted to show with this slide is that what I've learned so far is that although it doesn't matter how you start off in life, it is all about your drive and how much you want, what you want out of life. So right now I'm a 3rd year PhD student coming up on advancing to candidacy. And you know, this is one of the dreams that everybody's like, come on, you know where you came from, but it doesn't really matter. So some of the keys to success that I've learned during my path is that networking is so important. So here's a picture of a summer research experience that I did at NYU as an undergrad, and I was afforded this opportunity just by networking and talking to my professors, going to conferences, asking around, and then I ended up going to NYU as well as the Medical University of South Carolina to do 2 summer undergrad research projects. And I think the most pivotal moment in getting me into grad school was here at Abercrombie. So I presented some of the work that I got from my summer research institute at Abercrombie, which was a minority serving conference, a national conference. And there at Abercrombie, as I said, networking is key. I met somebody on the admissions committee for UCSC. We had a long conversation, sent in my application, and the rest is history, so to say. So I just want to emphasize the fact that it is really important to, you know, even when you're going to conferences or presenting your work even here, to, you know, network, to don't be afraid to showcase your work, and also work hard, and it's also who you know too. So networking is really important. I think that's what got me into grad school. So here's a picture of me graduating undergrad. So I started off undergrad at a private college where I did miserably. And then I ended up transferring into university in the South, where I just made a decision that if I wanted to go anywhere, I had to make great sacrifices. And here's a picture of my graduation. And you can get there too. It's just how bad do you want it? So here are all the people from my family who showed up to my graduation, and I'm sure they will for yours too. So here's a slide where I say, well-rounded is the only way to be. So of course it's tough, but also your collaborations, your networking is also contributes to your overall experience. So here are the other 2 graduate students in our lab. Hyman, he's here today, and Allison Conrad, she is not here today. But yeah, so one of the important things about being successful in lab is to foster an environment where you're comfortable and you're able to produce good work. So I'll take questions if anybody has any. Yes?
I remember reading about a year ago that they had discovered another arsenic-reducing organism in Mauna Lake, I thought. Do you know what came of that?
Oh, are you...
That it actually was reducing arsenic?
Yeah, so arsenic reduction is a really common practice. A lot of microbes have these AR or AB genes that we were talking about. But I think you're talking about the really big sensational story that came out. That was about a group of scientists claimed that they found microbes that are adding arsenic into the backbone of their DNA. And that, over the last year, many papers have come out refuting that. And they're still working on their project, but a lot of people are coming out with papers refuting their work.
I have another question.
Yeah.
You talked about you started your undergraduate studies at a private school, and then that didn't work for you. What was the problem? What wasn't there that you needed?
I wasn't motivated. So when I started off, I was a biology major. I knew I wanted to be in the sciences, but I felt like the opportunities weren't there. And my kind of flashback was, this is a waste of my time anyway. So what I did was I found a school where I felt like they could foster, my abilities, and then ended up performing really well at that school. So find what fits. Yes?
So you chose UC Santa Cruz for your graduate school. Were there other colleges or programs that you considered before attending UC Santa Cruz? And if so, why did you choose UC Santa Cruz?
Okay. So a school in New York called Stony Brook. I was also considering going into their program, but I was accepted into the pharmacology and more physiology type program. And I felt with UC Santa Cruz, it was more of an interdisciplinary sciences. So as I was saying before, I'm a molecular cell developmental biology major, but the science that I'm doing is environmental toxicology. So I felt like UCSC gave me the flexibility that I was looking for, because I didn't really know exactly what I wanted to dedicate myself to.
So I'll ask a question as a very, very non-biology based person. How do you isolate the CAMP as the
cyclic AMP?
Yes. How do you isolate that as the as what's doing the metabolism?
Okay. So that's a good question, because cyclic AMP does very many things, right? But the way we showed that cyclic AMP is directly involved in arsenic reduction is we knocked out all the genes that are important for producing cyclic AMP, which are the adenylate cyclase genes, and we found that it grew on oxygen fine, but it could no longer use arsenic. And then also we knocked out the cyclic AMP receptor protein, kept the cyclic AMP in the cell, and we found that when the receptor protein was gone, the arsenic reduction genes wouldn't turn on. So that's how we showed that this receptor protein, which is normally known to act as a transcription factor, which turns genes on and off. Once we knocked out that on-off switch, you couldn't turn the arsenic reduction genes on.
Are you mentioning to look at other genes, or are you just going to stay focused on the AR?
The AR, so it's pretty well established that those are the respiratory. So there are other genes. So the ones that are involved in detox, which is pumping arsenic out of the cell, and regulation of those genes. But I'm most interested in the genes that are turning this one on and off, because well, I didn't really make it clear, but in the presence of oxygen, so when it's breathing air, those genes are completely, like, they're not being turned on. So I want to know what other things in the cell are contributing to turning it off or keeping it off.
I'll ask one more question. What do you really like about being at UC Santa Cruz, and maybe what do you not prefer so much about, like the best and the worst or so?
The very best things for me at UC Santa Cruz is I feel like the faculty are really approachable. So when I have, let's say, a personal issue, they're willing to work with their students. So they're really dedicated to our success. So when I had some personal issues previously, they, worked with me to make sure that I was on the right path to success. And also the opportunities for collaboration. So you can come in as a certain type of student, but there's so many different programs that intertwine with each other. And I think that's really important to science right now anyway, to be able to relate what you're learning in your lab to a broader scientific community. And there's a lot of that at UCSC. Honestly, what I don't like so much is I think they're getting better at this, but diversity. So the amount of black students that are at the school is really low. I think for undergraduate, it is getting better for the last year. I've been there for 3 years. In the last year or so, I've noticed like an explosion of more diversity in the undergraduate programs, but it's still not as good in the grad programs yet. And that can be challenging when you feel like you need, in some instances, you would like to have somebody who understands you, you know what I'm saying? So that can be a problem sometimes, but like I said, the faculty do see the problem, they're trying to address it, and they are really dedicated to whatever you need to succeed.
Any more questions for me? Let's thank our first speaker. Marcello Quiros up next, and he's going to talk about his experience as a BS major at UCSD, molecular cell and developmental biology. He's a transfer from Ohlone College in Fremont, so a 2-year school as well, right?
Yeah.
All right. So let's welcome him.
So Pamela is my grad student, so she's my mentor. So she kind of went over pretty much the whole goal of the lab. I'm just going to reiterate a little bit about what we're doing. So I study arsenate reduction in Shmonella, which is the bacteria that we study in our lab. It's a model organism. So a little bit about arsenic. Arsenic occurs in many minerals, and it's usually found with in conjunction with sulfur and other metals such as iron. Arsenic is really poisonous to all kinds of life. Arsenic can go through a lot of redox conversions, which is very interesting and what we work a lot with in our lab. Arsenic is found a lot in groundwater wells, and this is a problem around the world. Pretty much wherever it's red, it's where it's a problem, and where you find arsenic in groundwater wells and stuff. So you can have that problem over here in the U.S. and down in Chile and over in Asia, and so it's a really big problem. Why is arsenic toxic? Arsenic disrupts ATP production through several mechanisms. And if you see over here, arsenate, it looks a lot like phosphate. So what you have is that it will compete with phosphate, and which can uncouple the electron transport chain, and also it can disrupt the protein structures because it can bind to methionine and cysteine. This is kind of what happens when you drink too much arsenic and it's found in your water. You get this disease called arsenicosis, which you end up getting kind of like gangrene on your feet and hands. So I mean that's really bad. A little bit about bacteria. This is it right here. The strain was isolated from a wooden pier because back in the day, what they would do with wood to preserve it is kind of, I guess, they would use like arsenic to help combat mold and bacteria. But this bacteria likes to grow on arsenic, so they found it on these wooden piers growing. So a little bit more about the groundwater contamination. The bacteria is found in soil, so it's hanging out in the dirt, and what it'll do is that it'll pretty much reduce the arsenate found in the soil and turn it into arsenite, which is a lot more toxic and water soluble. So you'll find this in the water, and then you find this form in the soil. So kind of like my job in the lab is to help find more genes that are associated with this process. So to do that, I do transposon mutagenesis. Pretty much what I do, I like to think of it as throwing out a fish net into the ocean and seeing what I get back. So I take the bacteria and I mutate it, and I screen for mutations, and hopefully I find something that's of interest. This is a little schematic on how I do that. Pretty much, I take two strains of bacteria. One of them is the one that we study, Shigella, and one of them is an E. coli strain with a plasmid of DNA. And what that'll do is that I mate them together, and I will create a mutation in the Shigella genome. After doing that, I screen for mutants that cannot grow anaerobically. So this would be a plate that would grow on my lab bench, and most of the time you would see full growth. And then I would check out a plate that's growing in an anaerobic chamber, and when I see no growth, like this little circle right here, that means there is a possible mutation in the gene that deals with arsenic reduction. And so that's pretty much what I'm working on in the lab. Here's a picture of my lab bench. It's pretty messy. And then this is a picture of bacterial plate, so you get an idea of what the bacteria looks like. It's not usually blue. This is actually this is like we standard blue. This is the lab. This is Oscar. This is me. This is Hyman. This is another undergrad. Whoops. This is Nate, Hyman, me, and then Oscar. That's messing around in the lab. And so, a little bit about my transfer experience. I graduated high school in 2008, and I went to Ohlone College, which is in Fremont. So to get an idea where that is over here, and we're like down here. So not too far. So how I started off community college, I had pretty much no idea what I wanted to study. So my first semester of community college, I took general ed courses like psychology, algebra 2, because I placed pretty low in math, and so I had to start, pretty much from the beginning. My second semester, I took a general chemistry course, which I really enjoyed. It was a really tough class, but I liked it. So from there, I went to talk to the counselor, and I was like, hey, I really like chemistry, and I really like biology and stuff. So they put me on a track to do that kind of coursework. But it took me 3 years to transfer because I didn't do so well in my first biology course. So so I spent a lot of time at community college. For a while, I felt like I wasn't going to transfer because, it was just taking a really long time. I didn't pass my first biology course, but I just kept on pushing through and taking all these classes, and then eventually I was in my final year, and I applied to pretty much all. I applied to UC Davis, Riverside, San Diego, Santa Cruz, Merced. No, I didn't apply to Merced. I applied, just to those schools. And I ended up getting into Davis, Riverside, and Santa Cruz. And I chose Santa Cruz because it was close to home. It's a really beautiful area, and I heard that they had really good research experiences and stuff, which I really wanted to do was going to research. So I mean, what I can say is just making sure to stay focused while you're in community college and stuff, and to make sure that every time you take your classes that you get all the stuff that you need to take. I know that can be frustrating. There were times where I couldn't take a class and I had to wait till the next semester, and that would kind of jumble up my plans and stuff. And also to make sure that before you transfer, you take all your prereq classes like chemistry, organic chemistry, because it's kind of a pain to have to take it at the UC level. And also, if you can, make friends who have the same career goals and the same educational goals, and who want to transfer and stuff, because you guys can help each other and talk about how you're going to reach your goals and stuff. So that's pretty much it. Any questions? Yeah.
When you were at the community college, did you have a chance to do any kind of internship or work in a lab on a project?
No, not at community college. I pretty much just took the classes and that's about it.
Do you see that as being an advantage if you have the opportunity? Or?
Oh yeah, definitely. I had a couple friends who they would do things at UC Berkeley. They would do research over there. Or they would come to Santa Cruz, and they definitely really liked that experience and benefited from it when it came time to apply to schools. It also helped, with their resume and everything. So, yeah, definitely, if you can do those kind of experiences.
I have a question.
Yeah.
So, if you go back to the slide where you have grown bacteria, so you mentioned that they were stained blue, but that wasn't typical. Yeah. So are you saying that you have some control plate and you have another plate and you compare them to see if there's something missing from the control plate, and that's how you know there's a mutation? Or how would you use this to decide if there's a mutation or so?
Okay, well, this plate was actually So the blue staining was actually to tell me that this reaction worked.
Okay.
But as far as what I was talking about before, going back to here, so when I look for mutation, I want to see something that won't grow anaerobically.
Okay.
So it's called replica plating. So I have a plate right here, and each of these dots corresponds to the same bacteria on the plate growing anaerobically.
Okay.
So if I do see growth, for example, oops. Yeah, like if I see growth on the plate growing on my bench top and on the same location, anaerobically I don't see a colony, then that means that there's something going on in that bacteria genetically.
Got it.
Yeah.
Why is that? So just to expand on it.
Yeah.
So the top plate is exposed to the air environment. So those bacteria, if I can correct me if I'm wrong, okay? But those are growing aerobically. So they use oxygen as their terminal electron acceptor. So the other plate, you put that in a chamber, we have an anaerobic chamber, and once that plate goes in there, there's no oxygen whatsoever. So what they do is they provide the bacteria with arsenate as a terminal electron acceptor. So any bacteria that are not able to grow in the absence of oxygen, and then you give them the arsenate, it should grow fine. But if this transposon jump in some gene that is important or regulating the metabolism, now you are inhibiting that, and now you can't breathe on arsenic, which you should be able to.
Okay.
Yeah.
So you just change the conditions of Peru. That's pretty much what you do then in that case? You cut out the oxygen and you give it arsenic to see if it will survive?
Yeah, because he was looking at the arsenic metabolism. So that's why his control is the oxygen. You want to make sure they're going fine, they're healthy. But then your real test is testing them under the arsenic. Because you can't give them arsenic and then grow them out in the environment, because oxygen is a way better terminal electron acceptor molecule. You can just tear off the electrons from the electron transport chain, and then arsenic, it just can't compete with oxygen. So that's why you have to put it in a different environment. If there's no oxygen, give it some arsenic.
So what happens with those organisms that do not grow under the anaerobic condition?
Well, in this case, it's a replica, so that all the bacteria up there are all the same. They're Shionella 3. So there's just a bunch of them, but each individual well potentially has been interrupted in its genome somewhere at a different place by this transposon. So that's why he's kind of interested to see which is the gene that this transposon jumps into and interrupts this arsenic metabolism.
So, can I ask you a question?
Yes.
So, Marcelo, I think what she was asking is, what do you do if you see one that didn't grow on arsenic?
Yeah, so one that didn't grow on arsenic, I would plate it again and aerobically to make sure that it won't grow. And then I go back to the replica plate growing on my bench top, and I pick that, and then I extract the DNA. Well, first of all, I would do a growth curve to make sure that it's not growing anaerobically, and then compare it to the wild type, which will grow anaerobically because there's no mutations then.
What's a growth curve?
A growth curve is where you would grow it in a tube anaerobically in a culture, and you would pretty much let it grow for about 12 hours, and every hour or so you would take a measurement using a spectrophotometer to see how much bacteria there is growing inside the culture.
So,
No, yeah, so I mean that's pretty much a growth curve. So then after that, once I confirm that it's not growing, then I would extract the DNA from the bacteria, and then and then I would send it off to be sequenced to find out which gene was mutated by the transposon.
It's pretty cool.
So yeah, so how long did it take you to like become comfortable using the spectrophotometer and extracting DNA? And I mean,
Yeah, it took a while getting comfortable with that because it's really complex and there's like a big protocol you have to follow and
Sure.
and everything, and I made a ton of mistakes.
Okay.
And I mean, you can make mistakes at step one, and you know, so.
I'm just
but-
asking because as a student in the audience myself, I mean, I know physics. I don't really know biology, so I'm sitting here kind of intimidated by this. I would think, how many semesters does it take
I've been
to get
working
to your?
since last winter, and I've been pretty much doing the same project up until now. And so far, I've found 2 mutants. I've done this process, and then I've found 2 mutants, and one of them was actually something of interest. So, but I've probably done this process like, I don't know, dozens of times.
You've made a few mistakes.
Yeah.
Fair enough.
So was this your first choice to enter this lab, or did you kind of just land on this lab?
I kind of just like landed in this lab. I'm part of the IMSE program at school, which is a STEM program, and I went to talk to the director, and I was like, hey, I want to do research, you know, and then she's like, okay, we can help you out, and I was looking through this abstract journal from a research symposium. So I was going through it and I was like, arsenic reduction, hey, that's interesting. And I started reading more about it. And then the next week I made an appointment with Chad, who runs the lab. And you know, I made an appointment with him and had an interview, and that's when I met Pam for the first time. And then I had to like interview and apply, and then I got into the lab.
So you're doing your classes and spending time in the lab too?
Yeah,
Is that like,
yeah, that's definitely.
How's that
what's it
work?
like balancing that?
Yeah.
Yeah, it's definitely a lot of work because during the school year you should be putting around 10 to 20 hours a week in lab, and then of course, you know, you're taking anywhere from 12 to like 16 units of classes. So usually when I'm not in class, I'm in lab, and then of course like study time. But it's definitely hard to jumble, you know, studying lab and class, all that stuff.
Do you want to go on and do a graduate program, and if so, do you think it'll be at Santa Cruz?
At Santa Cruz, I don't think I would want to stay in Santa Cruz for graduate school, no. But yeah, so but I would actually like to go into cancer research. But yeah, so right now I'm going to start working on applying to grad school and everything. So.
Any other questions from the topic?
Why cancer research?
Because I really like studying about cancer, and I'm taking a couple courses that have really gone into detail about the mechanism of cancer, and that's something that I find really interesting. So I'd like to learn more about it and do research on it.
Well, very good. So let's. So we are next welcoming Ana Hernandez, who's a graduate student in the Department of Microbiology and Environmental Toxicology, also studying arsenic metabolism. Okay. So yeah, so let's welcome him. And I'm going to be obnoxious and take a picture, because that's what I do.
Okay, so my name is Jaime, and I'm gonna do kind of what we're all doing, talk a little bit about research, and then a little bit about myself. So my title is Engineering Microbes to Detect Arsenic in the Environment. Unlike Pam and Marcel's work, I'm a little bit more interested in the environmental work and seeing how I can apply utilize this microbe and use its genetics to figure out more about the arsenic. How is it cycling in the environment? How is it going from the soil to the water? And we obviously know that some of these genes are important, but a lot of the work that's also out there suggests that there's other processes that affect arsenic cycling in the environment, and it's not just this microbe. Other people say, oh, well, there's other molecules that are inhibiting or kind of pushing arsenic from the sediment to the water, and those are also a big cause. This is a little bit of my project, and I don't know. I also, it's more applicable, not so much here to the U.S. I'll show the little graph Marcelo showed, a map of arsenic around the world. And we have a big problem here in California, but we are actually, we can deal with it because we have the facilities to clean our water systems. But other places in third world countries are not as fortunate. So if by designing these microbes, we can, other third world countries or people that have this arsenic problem can utilize these microbes for arsenic sensing in the environment. So arsenic it's pretty cool chemical. It has no color, it has no odor, and if people ever come across arsenic when they drink them in the water, it doesn't taste like anything. So it's like a perfect toxin. So again, Marcelo mentioned that consumption of arsenic can cause this lesions and diseases such as arsenicosis. I don't know how to work this. I use some laser.
Okay, so that works.
You could also use the arrows in the keyboard and that.
Right. But I promised that they could use this, and
I'll use the nice.
okay. So
I'll use the nice.
okay.
I got it. So I'll give you guys a little bit about toxicity, just to introduce, just to compare, see how toxic or levels of arsenic that are found in the environment. The EPA, which is the Environmental Protection Agency, has a standard about 10 micrograms per liter. And again, in other places like Southeast Asia, Bangladesh, this exceeds about 100 times as much. So it's about 1,000 micrograms per liter. So that's here. So going back to arsenic, as we mentioned again, arsenic is right below phosphorus, so it mimics it. When we are producing energy, phosphate really, it's an analogous to arsenic. So arsenic can inhibit various things. Energy production, which is ATP. ATP molecules need phosphorus to have this energy. It also, when we're building going back to the paper that you were mentioning, arsenate or phosphate actually is another molecule that binds to the backbone of DNA, and then you have DNA and then your bases. So that was the whole paper that they were talking about. With when you don't give the microbe any phosphorus and you give it arsenate, it mimics it so well that it could uptake arsenate and build DNA with this arsenate versus phosphorus. But yeah, like Pam said, that was kind of, there's more research being done that is not, says the opposite. It really can't happen. Not very favorable. So yeah, so Marcelo mentioned where arsenic can be found. The green areas are geothermal environments. So these are environments that are, there's a couple in California, Hot Creek, Mono Lake. So they're not really very good here, but there's like I said, we have a big arsenic problem there, and there's the concentrations do exceed the EPA standard levels. But fortunately, we have systems that we can purify the arsenic, and for us, it's not a problem because we can get most of the arsenic out of our water. But those are all sources that are coming from geothermal environments. So yeah, you go out there and you see these ponds of boiling water and a bunch of arsenic that's coming from underground. We also have the other areas, the circles in there, they're affected by mining operations. In Chile, there's a few of those too down here, and around Bangladesh also is affected by groundwater. So yeah, these are all the sources of arsenic throughout the world. And in my project, I want to generate these microbes to better understand that cycling. How is the arsenic going from a sediment to the water system, and then being, once it's in the water, that really affects people that uptake the water for drinking purposes or irrigation. So what I hopefully want to do, I'm going to come back to this image here, is engineer a microbe, but insert this GFP molecule, so it's a green fluorescent protein, downstream or in front of the genes that we already know that are responsible for arsenic metabolism. So anytime that microbe is in this environment where there's no oxygen and it's forced to either breathe or eat arsenic, we have this microbe that will, as soon as it's doing its metabolism, it'll glow green, and we'll be able to detect. Hopefully, the idea is where the arsenic is coming from, and then also concentration. So if the fluorescence is low, we'll say, oh, there's very low concentrations, and very high fluorescence, there's a lot of arsenic that is being mobilized. So we talked about some of the genes that are important in arsenic metabolism. The ones in green are these systems that, so this shows two things. The top, the mineral when it comes in contact to the microbe, and then this here would be a mineral that is iron with some arsenic, and then here this is a microbe. So this is their membrane, and in the lower, in the cytoplasmic membrane, you have these proteins that a lot of these microbes have this mechanism, but in this does not allow for respiration. This is only for detoxification. So when arsenic comes in, again, it mimics phosphate, so it comes through this phosphate pumps, and then once it's here, this molecule can recognize it and then change it from arsenic 5 to 3, and then this gets pumped out of the cell and into the environment, and then the microbe is healthy, it's fine. It doesn't have to deal with any of this inorganic arsenic that's in the cell. So that's the detoxification mechanisms. A lot of aerobic, anaerobic microorganisms have this type of system, and it allows them to survive in high arsenic concentrations. We also have the arsenic reductase, and this is in the periplasmic of the microbe, and it does similar the same. So when you have arsenate, it can breathe on arsenate, so going from arsenate to arsenite. So the electrons are coming from here, going through the electron transport chain, being donated to cytochromes, and then finally to this molecule, and then this process, again, only in the absence of oxygen, you have this microbe that can breathe on potentially, which is a toxic metal. Oscar will tell you guys a little bit about another proteins that these are iron, involved in iron respiration. So when there is either no oxygen or no arsenate, then this microbe can utilize these proteins to breathe on these soluble iron minerals. So this is also some of the research that was pretty new up to maybe 10 years ago. They're like, how can these microbes uptake these electrons from solid minerals. Because normally all the compounds that are either diffused and have to get all the way down here. But in this case, this microbe has proteins that are outside of the membrane and then make contact with iron 3 and then reduce that to iron 2. And then that's how they donate their electrons. So Oscar will probably talk a little bit about that. So in lab, currently I have generated what we call them, Marcelo was talking about knockouts, so disrupting genes, and in this case, I'm doing a knock-in. So I am inserting this GFP molecule again in downstream of this ARR and ARC. And so far, I've gotten these two, and our Oscar, with Oscar's help, will probably get our last one. And once we have these strains, we'll be able to put them into this cool system that has been designed by a professor in Stanford University. And it's a little complex, but hopefully I do a pretty good job in explaining this process. So out in the environment, we have soil aggregates. And as you can see, you have clumps of soil that at times they're really compact, and there's other areas where they're very hollow. And this is going to be affected throughout the year. When there's rain, there's going to be water, there might be oxygen, and then when the seasons are dry, then there'll be no oxygen within these gaps here. So depending on the environment, the microbe is either going to respirate oxygen if it's around, and when there's no oxygen, it's going to go anaerobic and then breathe on either iron or arsenic. So what's shown so far is the little spheres here. So these spheres we synthesize them in lab. We make some ferric chloride with an acid, and then we have our ferric hydride, so it's an iron 3 compound, and then we mix that with sand, some quartz, just to get more, build a surface area, and then we use this aggro, so it's kind of like gelatin looking like, and then we have these nice little spheres that we can build our spheres with. And once we have those, the idea is to put these fluorescent microbes in there, and then we can incubate them in a chamber like this. And then the idea is that it's anaerobic. So again, it has to breathe on arsenic, and then when it's breathing on arsenic, it's utilizing those genes that I talked about, and we should see fluorescence. If it's breathing, it should also be detoxifying the arsenic, so we should see some of that. So we would incubate these microbes. So there's 3 different strains that I talked about: the ARR, the ARS, and then the iron. So we would incubate them separately, and we would see fluorescence like this in this aggregate as time goes by, because either the microbe is detoxifying or because the microbe is breathing on arsenic. And we could either see it through some type of confocal microscopy at the fluorescent, or there's also these cool machines that we can actually extract the microbes from here, and then it's sort them through fluorescence. So we can do it directly on the aggregate, expose this to some type of microscope, or extract the microbes from here and then sort them through fluorescence. So we want to get a sense of, again, where are these microbes in the environment, and how much are they playing a role in arsenic mobility, going from the aggregate, which is mainly arsenate, and then into the water, which is arsenite. It's more water soluble. So I think we forgot to mention that a little bit, how arsenate is more bound to soils, and once it gets reduced, then arsenite becomes water soluble, and that's how it's dominant in your water source. So background. So unlike my colleagues here, I was after high school, I was not really focused, and school was not my priority. I had at one point like 3 jobs. So I was working like 7 days a week, and it was mainly just for I don't know what I was thinking, but I was living back at home, so I was not paying any rent. All the money that I was making was, just bought a car, I don't know what I was thinking again. So I was not going to college. I signed up, but then I never attended class, so my first 2 years were just a waste of time because I never went to class. But after working like a year and a half, 7 days a week, I just realized that that was not a path that I wanted to keep going and doing. So I had to force myself to enroll back into Cabrillo College. So these are some of the jobs that I had. I worked at Stagnero's over in the wharf, cleaning some fish. It was pretty nasty, very smelly. And then during the morning from like 2 to 7, I would just work at UPS and part-time, and they had like great benefits. So yeah, it was great. But I was not in school. So after I got tired of that, I decided to go back to Cabrillo College, spend a lot of times in the library, and here in this MESA, I believe you guys have a MESA program here too. So that was a lot of help. All the people that I met in MESA were super friendly, and we always stayed up late to do physics, calculus, whatever we needed to do. And so after I got on track, I started acing all my classes and got into biology, and then that's when my interest in biology again arose. I was like, wow, there's all these crazy molecules that we can't even see that are like doing all these cool things, and enzymes and are important in cancer and all this stuff. So that was really awesome. And another Access was a program that facilitated and even kept me going, because Access, I think, you guys have Access here too. So if you apply and you get in, you get to go to university, UCSC, and then work in a lab there. And when I was up there, I worked my first year in the lab that I'm doing my graduate program now. So this is the Saltykov lab. And I was doing a lot of environmental work, which I didn't like, but at least I got a sense of what was out there. I didn't even know this type of environment was out there for me, like doing research. I was like, what is that? So I didn't know about that. So I found out through Access. And the second year, right before I transferred, I did Access again, but then I worked in Dr. Camp's lab, and he did a lot of DNA repair, so a lot of like cancer-related stuff. And that was really interesting to me. So by the time that I got to UCSC, I took a lot of cancer courses. I already knew what research was about, and I just I applied to other programs that we kind of mentioned, that are minority programs that allows us to get into a lab and keep doing research while you're still studying. So when I applied to the university, I actually got into UC Davis and UCSC. There was another school that I applied. I forgot which one it was. I think it was San Diego. But at the time, I was having my first baby. Well, my wife was. So I was kind of restricted to staying at UCSC. So I stayed at UCSC and I transferred there. And I did my undergraduate there. I did a molecular cell development. And when I was finishing there, I applied to there, and then also another cancer institution down south in LA. But we have such a great support from, I have family in Santa Cruz, and my wife does too. So we were lucky and fortunate to stick around and got accepted to graduate school, and I kept, so that's what I did there. So it's great to do these summer internships, because just this last week, Dr. Camps, he sent me an email, and he was like, oh yeah, you remember all that stuff you did and all those library that you generated? I was like, oh yeah. He's like, yeah, we're gonna make a book and you're gonna be a co-author on it. I was like, oh wow, that's great. Cause I was like, I didn't think that was gonna go anywhere because it was a lot of work. It was kind of like what Marcelo was doing, just trying to catch a fish in the net. So it was a big, if we got lucky, we were gonna get somewhere. If not, it was just a lot of like random mutagenesis. But anyway, we're not. So that was pretty cool. So I'm gonna be a co-author in a book. So it pays off. You guys, when you guys do these internships, you guys have like a co-author book, or sometimes papers get published. A lot of the times you guys get a letter of recommendation, which help a lot to transferring or applying for jobs out there. So it's a great thing. You guys should take advantage of that. Let's see so this is our sketch of our building, which is fairly new. This is a PSB, physical science building, and we're over here on this side of the building. And now there's a new building down here, which is the biomedical research building, and it's it's pretty great. It's our science department is expanding. You guys should attend UCSC if you guys are into biology, chemistry, or physics. We have a great physics department too. So this is our lab, and I want to finish with some with my funding, IMSD, and our PI is here, Chad Saltikov, Marcelo, and our crew eating some, what was this? Indian food? Thai food. Yeah. So, yeah. Any questions?
Questions? So did you land on this lab, or did you actually want to be in this lab?
For my first time or my second time?
Second time.
My second time, yes. My first time that I actually applied there, before transferring, I was a soil and water science major. So I've always been out like in the environment. I was going to pursue also like environmental engineering or agriculture, something like that. I definitely wanted to study more about these toxins and how microbes play a role in the environment and how they cycle, not just arsenic, but other pathogens. So that was my top choice. So I ended up working out.
So you mentioned this sophisticated system where you're going to test or you're able to like sort of assess what the microbes are doing and if there's arsenic respiration going on, right? Designed by the professor at Stanford. So what's really special about that system or so?
So again, there's nobody has really shown also how much done any measure any of these genes. They just know, oh, okay, well, if we do these, they do various things. They do like columns. Marcelo showed a column. So if we put a microbe in there with arsenic, they show that the arsenic goes from the sediment to the water, and they're like, okay, well maybe the microbe is playing a big role, but nobody's ever done the actual like how much are genes their role in the mobility of arsenic. So that's my kind of interest, and the big goal again is just to see and provide other ways that we can measure arsenic. So normally we measure arsenic through like ICP-MS, which is very cost effective, and in third world countries we can't. They're not fortunate to have those types of systems. So if we have microbes that can detect arsenic in the environment, we could by fluorescence. So you don't even have to measure. So it would cost a lot. So you get your microbes, you take it to the lab, you extract DNA, and then after that, you have to do this qPCR just to measure the mRNA, and then from there, so all these multiple steps versus just like going out there and seeing the microbe visually through this protein if these genes are activated or not activated. And I mentioned the fluorescence kind of plays a role. If there's low fluorescence, there's very low arsenic mobility, and then a lot of fluorescence, there's a lot of arsenic mobility, more respiration. So just a way to detect and kind of arsenic out.
So the intensity of the fluorescence would indicate the amount of arsenic.
Concentration
Yeah.
increase.
You did mention also something about he does the take out and you do the take in.
Mm-hmm. Yeah, so we're trying to insert this fluorescent molecule in front of the gene. So that way, when the gene is transcribed, it's made, then you have this, it makes this gene, and then right after that, this protein is there, so it will make also this gene. And once that is made, then it get translated into a protein that will fluoresce green. Yeah. Yeah.
So I saw in the picture with the fluorescence, it's like zoomed in, real small. So is that something you can only see under like a microscope, or will you actually see the soil glowing?
Yeah, I'm trying to. So this is the hard part of the project. Again, nobody has ever measured, or it's really hard to get to see fluorescence in soils because the minerals themselves give out a big readout, and you can't really distinguish. It's not as clear as this. So this is just like microbes on a slide. But when you're looking at a rock or a mineral, there's all these things like a big chunks fluorescing right there. Is that a big clump of microbes, or is that minerals? So that's what I'm kind of working right now to try to get a better way to image these microbes. But confocal microscopy is a way that we should be able to be. It's sensitive enough to detect this fluorescence, but we would not be able to see this nice image like this here. Yeah. The other way was I was saying is we instead of doing that, then we just extract the microbe and then isolate the bacteria itself, and then put it through some machine which can sort the fluorescence. And since this is a green fluorescence, we know the emission and excitation wavelength, and we can just say, okay, just collect only these ones, and then we can see again, are they fluorescing a lot, a little, how many bacteria are there in a given surface area, and just do more close to that.
So you had to clean it up a lot.
Yeah, it's a lot more work that way. Yeah.
The fluorescence is dependent on an anaerobic condition?
So because we're I'm doing 3 different strains, that depends. So if on the stuff that this is the detoxification mechanism. If once arsenic is there, fluorescence will always exist, aerobic or anaerobic. The microbe doesn't care. It's just like, oh, I don't want arsenic. I just need to get it out of myself. And once it does that, it transcribes this gene that it's the genes that are important in detoxification. So then fluorescence will be present there also. So in that case, there'll be fluorescent, aerobic and anaerobic. And then in this case, only when we grow our sample anaerobically, we'll see that. So yeah, this is really again interesting because we can put our microbe theoretically out in the environment, and it always be on aerobically or anaerobically, and we can kind of get measurements depending on. Well, first we have to quantitate the gene transcription and through fluorescence kind of, and get a sense if there's a correlation there. And if there's a correlation there, then we say, okay, fluorescence does equal gene transcription and arsenic mobility. These are the concentrations that are being released or mobilized. Yeah.
So how far away are you in terms of like a timeline from being able to help third world countries mitigate arsenic? Like 10 years? Is that even a question that you guys are asking right now?
So theoretically, it's once these strains are made, which they are, a couple of them are, we have to again go back to doing like what Marcel was saying, growth curves, make sure that the microbes growing healthy, that this knock-in did not disturb anything, and that they're able to grow fine. And then get a sense of if the gene transcription correlates with fluorescence, and we can measure, okay, if there's this amount of concentration of arsenic, we get this amount of readout. So if that, then we could ideally be collaborating with other engineers that can design either some type of devices that we can have the microbe there, and then you just shoot a water sample onto your microbe, and you can just get a sense of how much it fluoresces and say, okay, there's really low concentration of arsenic, or there's not. So there is systems like this out there. They're just not as very sensitive when it comes to very low arsenic concentrations. So that is a challenge also, just to see if we can get these microbes to glow at very low concentrations, because those are the concentrations that are measured out in the field. So so that I haven't even tested for that, how much varying my concentration. I've just done the knock-in, GFP's there. I gave it a bunch of arsenic and they're fluorescent. So now I have to go back and see what's the limit. Yeah.
So it's more about being able to detect the concentration.
Yeah.
Have you had an opportunity in your program to go to a third world country and see sites or talk to people in situations that...
This is my first year, so I just finished my first year. I got a chance, just to go out to the field where these microbes are isolated here in southeast California, Mono Lake, Hot Creek, but I haven't made it to the field yet. We are collaborating with another professor that's coming from India, so maybe after that he comes here, then I go over there, something like that, but I haven't gotten a chance yet.
Well, let's So our final speaker today, Oscar Fernandez, he's also in the Health Lab. He's going to talk about his research and his personal journey from CSU Fullerton to and his goals. So pursuing a PhD in environmental toxicology or biological chemistry. So let's open.
Hopefully you guys are not tired of hearing about arsenic, but it will be a few more minutes. So yeah, my name is Oscar Fernandez. I transferred to UC Santa Cruz, and I'll talk about that a little bit more. I'm a Jaime's undergraduate, and I've been working in the Salty Cup lab for 9 months now. So yeah, so here we go. So Jaime mentioned already that there's bacteria that breeds in different environments, like when there's arsenic, when there's no arsenic, when there's oxygen, when there's no oxygen. But this bacteria can also use iron to breathe. As we breathe in, let's say, oxygen, this bacteria is breathing. In the case that I'm looking at, it's iron. So iron is the fourth most common compound in the earth crust. So it's ubiquitous. It can be found everywhere. We have it throughout our body. You know, a slide of arsenic and lead. We'll just focus on the arsenic part. So there's various ways to ingest arsenic. You can either breathe it, you could go in through your skin, or you can via orally. And it does affect various things in your system, and can even lead to cancer. So again, I'm not trying to gross you guys out, but this is the reality of why arsenic is a threat, I guess, to the human health. So again, as I was talking, my project involves more on this gene, this cluster of genes called MTR. And what it does is a lot of bacteria that reduce metals conserve these genes for reducing reducing metal. Some of them can reduce iron or manganese. In my case, we're looking at iron reduction. And as I mentioned, we found in the environment that when arsenic is bound to a type of iron, when we have this bacteria, like in our case, our model organism is called Shigella, this dissimilatory metal reducing bacteria disintegrates or breaks down the iron and the arsenic so that way they can uptake what they need in order to survive. So once they break these compounds, arsenic is no longer insoluble in water, so now it's soluble in water, and it can make its way into like water flows or like rivers or like wells and potentially contaminate whatever source of water there is. So this is what my project entitles. Right now I'm helping Jaime construct this bacteria strain or reporter strain. So what I do is, we take the same procedures of knocking in a gene inside a region within the genome of the Shigella. So what I've tried to do for this past months is to insert this GFP, this green fluorescent protein, just downstream or just adjacent to the genes that are called MTR that are essential for reducing like iron. So at this moment we're still trying to push in the last piece on the puzzle, and then we can insert it into this aggregates, as Jaime showed a picture of the aggregates, how we place it in the aggregates, and we put it in these conditions, whether it's aerobic or anaerobic, and we leave it for a month, over a month to to do the magic, I guess. And then we just cut it and take really, really fine slices and test it, how it mobilizes. Once, I guess, some of the future work that we could do after we get this strain is making sensors, as Hannah said, to detect whether there's high levels either of iron or arsenic, and we can get some of this work done. I guess here's in writing what I'm doing in my research. So I'm just gonna talk a little bit about me. I was born and raised in Mexico, and I came to the States when I was 14 years old. I attended South Hills High, not knowing a word of English. I was thrown in this 90% Caucasian environment, and I had to pick up English pretty fast because it was something. And then I was homeschooled for my last year and a half of high school because of health conditions. But after, I guess, I got better, I continued my schooling, my education at Citrus College, which is located near Pasadena. I traveled to both Pasadena City College and Citrus College, so you guys know what that is. You can't find a class here, you have to go somewhere else and try to find the classes that you need in order to graduate or transfer. Through Citrus, one of my professors once asked me if I was interested in research, but at first, I really wasn't. I wanted to become a medical doctor. So she's like, why don't you apply for this internship? So I applied for this internship, and it's called the REU, the Research Experience for Undergraduates. There you go. So it's a NSF funding program, I guess. So I went to Cal State Fullerton during the summer of 2008 and 2009, and I worked in Dr. Dratt's lab where he focused on the metastasis of tumors to a primary site to a secondary site. So that was actually pretty cool that, and then in community college, it's pretty hard to like look for these internships, but thanks to my professor, she asked me to apply, and I did, so I'm really thankful for that. So I transferred to UCSC in fall 2009, and as a biomedical engineering, but I didn't really want to go into industry. I wanted to do more research since those two research experience really got to me. Like I really just wanted to focus on research. So so when I applied to the UCs, I guess I got accepted UC San Diego, UC Santa Barbara. I got a full ride to UC Merced and UC Davis and UC Santa Cruz. But one of my professors said, the same professor that told me about the RU, that if I wanted to do research, I should apply to UC Santa Cruz, and that's what I did. I should be receiving my my bachelor's degree in chemistry with the biochemistry concentration this upcoming spring. Yeah, so that's a little bit about me.
Do we have questions? So,
Okay, so just college, were you going by the semester?
Yeah.
Okay, so how's it going from the semester to the quarter system?
Awful. Awful. Yeah. The amounts of reading that you're expected to do, it's incredible. Yeah, it's like nowhere near comparison. You will have to read like two chapters, 3 chapters a week for one class. Now add 3 classes, so that's like a couple. But it was different. It was different. Your expectations, what the UC demands of you it's more than I guess what I had in my community college. But you get it right away. Like you get it right away. You start. There's no room for slack. So don't worry about like not fitting in or not not trying to do well in it. There's no room for slack because there's so much. It's so fast paced that you really don't have too much time to be wasting. So as soon as you get in there, you realize how like hardcore it is. So you just jump in and assimilate. There's no other way, to be honest. But the amount of reading is awful.
It's about 2 months, right? So each quarter is about 2 months.
Yeah, it's 10 weeks. So your first week is, it's not reading syllabus no more. First day of class is like, okay, open your books in chapter whatever and go on. So yeah, that was one of the biggest. And still, UC Santa Cruz is still a really warm place, whereas other UCs, they're like, the professors, you can even talk to them. Here at UC Santa Cruz, I felt extremely welcome. It was like, you assimilate, or remind me a lot of my community college, that I could just drop into professor's hours, and if I didn't find them, I would go to around campus, and I'll find them around, and then I'll ask them questions. UC Santa Cruz is somewhat the same way. Classes are big, they're 200 students, but office hours are always there, and then pretty much professors live in their labs. So, if you really need a question answered, you just knock in their labs and be like, hey, Professor so-and-so, this is what's going on. And they actually like it, they actually like that you bug them. Yeah.
how did you end up working on the research project that you're working on?
So I applied, since I transferred in the fall 2009, winter 2010, I applied for the internship that I guess pretty much will have. And I got in. So during the summer, what they do is if you're already working in a lab, you stick with the lab, you work full time in the lab, 40 hours a week, what is expected of you, and you just work. If you don't have a lab, what they do is they put you in these rotations where you rotate in 6 different labs, chemistry, MCD, whatever it is. You just rotate and kind of learn those skills that are necessary for whatever lab you're gonna apply to. How I applied to the Celtic lab, I talked to Jaime, I know that he was working in there, and I go, hey, I kind of like, what they're doing out there. I'm not too familiar what it is. Do you have any like papers, or you like maybe suggest? He's like, just look in the webpage and that's it. So I looked in the webpage and I read one of the 2 of the papers that the PI published, and then I went to. He's like, okay, I need to talk to my PI, and he set up an appointment. I had my interview, and then after I got the okay, then I started working, and how we have.
You mentioned you were thinking about being a doctor. Do you find that your current research kind of scratches the itch of what you
I'm
were?
no longer looking to be a medical
Right.
doctor. At first, I thought that I wanted to be a doctor. I wanted just to everybody has the hopes of like, oh, I'm gonna help people,
Sure.
I want to go like this and that. But I think that the science spoke, I guess, louder to me, and I really, really love being in the lab, just in the lab setting. I think that's what I feel more comfortable with. And it's not that you're not going to help less people,
Sure.
because I guess even if we get to Jaime's case, making that reporter strength, can help not only the one person that was in the bedside, it can help, countries and, the whole nation, I guess.
Sure.
So, yeah.
Just curious, what is the generation time on the
Shonella?
Yeah, is it slow or is it?
Oh, it grows overnight. It grows overnight, and it's stinky. Like you leave it in
It's got the same generation time as the E. coli. So
20 minutes.
20 minutes.
Yeah, but it really stinks.
Well, let's go ahead and thank Oscar, and let's thank all of our speakers tonight. So this concludes this installment of our.
Is it all over?
It's for today. Yes, so this concludes this particular meeting of our STEM Diversity Physical Series, but we'll be back in a couple of weeks.